Good morning, everyone, and welcome to today's webinar, Accelerating Single-Cell Genomic Discovery with AI, NVIDIA, and CodeOcean. I'm Sharon, I'm the VP of Operations at CodeOcean, and I'm thrilled that you all joined us today. Before we get started, we just have a few housekeeping items. First, the session is being recorded, and we'll send you the recording after the event. Second, we'd love to get some questions from the audience today. Please feel free to submit your questions anytime throughout the webinar using the chat on the right-hand side, and we'll answer them towards the end of the presentation. I'm delighted to introduce today's speaker, Emily So. Emily is a solution scientist here at CodeOcean. She focuses on applications of machine learning in bioinformatics and GPU acceleration, and has prepared a great presentation for us today on how GPU accelerated workflows can drive fast and accurate single-cell genomic discovery. Let's talk a bit about what we're going to cover today. We're going to discuss current AI and machine learning use in single-cell genomics, advantages and challenges. We'll discuss using GPU acceleration to scale and speed processing and to improve the accuracy of outcomes for large single-cell genomic datasets. We'll also cover how to remove productivity and computing limitations using your existing code and packages and CodeOcean compute capsules. And lastly, we'll wrap up with a live demo of how it's done in compute capsules that you can then use on your own. Emily, thank you so much for joining us today, and I'll pass it over to you.
Thank you so much, Sharon. So before we start diving into really the content of the webinar, I think it's important to address why GPUs are so important in single-cell analysis, why it's really an exciting advancement, and why it's important to focus on it in bioinformatics. So as you can see on the image that I have here, there is a significant improvement in runtime when using a GPU for any type of single-cell analysis. You can see on the left here that it's pretty typical methods such as TSNE, k-means clustering, and a lot of pre-processing, but the acceleration factor is pretty large. You can accelerate a process by almost 20 times using a GPU compared to a typical CPU. And as a computational biologist, that's really interesting, because these types of processes that you're performing use quite large data sets. And so to be able to do it in this short amount of time is really exciting. Not only that, you're saving almost a third of the cost when you're using a GPU compared to a CPU, which is also really beneficial, which we'll maybe talk about later. So now that we've kind of introduced the advancements that have occurred with GPU acceleration, I thought we would do a little bit of an audience poll just to see everybody's knowledge about GPU acceleration, if you've experienced it, and other computational challenges you may be facing currently in your research.
Okay, I am starting to see some answers coming in. Emily, I think you'd be surprised with what we see here.
Yeah, I am pretty surprised. I think that's really interesting that there are quite a lot of people who are actually saying not sure, more so than yes or plan in the future. And I hope those who are saying not sure, I hope we can convince you that GPU acceleration is in fact pretty remarkable and important in single-cell genomics. That's really interesting.
Okay, let me share with everybody, because right now, Emily, you're the only one who can see this. Okay.
Yeah, it's really interesting.
Right, so if you're not sure, please write to us on chat. Why are you not sure? What does it mean? If you can explain a little bit more some of the challenges that you have in mind, we think that will be great to discuss more and think about it a little more. If you're also planning to use GPU acceleration in the future, it will be interesting to hear what's preventing you from doing it today. I'm going to continue to the next poll. And we'd love to hear from you with your current workflows. What challenges are you experiencing? Please feel free to choose more than one. Interesting. I'll give it a few more seconds because I still see responses coming in. Okay. I'm going to close it. Emily, thoughts?
That's also, again, a really, really interesting result. I find it super interesting that quite a few percentage of people are facing the challenge of sharing analysis with biologists, which is actually pretty common in computational biology. I feel like at the heart of the field is translation to other disciplines, so that it is really interesting. And I think we're going to touch on a lot of the issues that people are facing today.
And Emily, do you want to try and take over again?
Yeah, I think we're having just a little issue right now.
There we go.
Okay. So now that we've kind of talked about GPU acceleration, it's important to talk about all the components that kind of led up to the advancements that have occurred. So let's go back kind of to the beginning and work our way back up to implementing GPU acceleration. So it's important to first discuss workflows in single-cell genomics. Single-cell genomics kind of came to prominence by its ability to explore genome-wide expression data at the single-cell level. And this increase in resolution is obviously much better for analysis. It's much better and produces much clearer results when you have DNA and RNA genetic content for each individual cell in a data set. And this led to multiple analyses that could now be performed. One of these is the clustering to be able to identify and characterize cell populations. So as you can see in the image below, we kind of have an example workflow laid out for you. It involves cell isolation into a data set of containing single cells. Then that leads to gene expression, which usually results in producing a matrix in which the expression of each gene is kind of labeled with a different color depending on its magnitude. And then it kind of leads to this kind of clustering analysis in which each different color is either a cell type or a cell marker. And this can be combined with other data for future applications, such as identifying targets for therapeutics or identifying potential patterns that are related to genetic disorders. There's a lot of applications for this kind of framework of analysis. And over time, as single cell data sets have continued to grow, that's kind of been the crux of the computational challenges that researchers are facing. And to provide a solution for this type of challenge, the use of AI and machine learning in this type of clustering has quickly become the gold standard. The reason that AI and machine learning was first implemented to solve the problem of single-cell datasets growing is by its ability to apply scalable algorithms to a large dataset in order to produce clear interactive results. And these methods that allow for scalability is dimensionality reduction, which involves taking a large dataset and scaling it down to a smaller one for clearer resolution, batch effect integration, which entails accounting for any biological variation, and unsupervised clustering, which involves learning patterns in the data set as the algorithm progresses. And when you're able to implement these types of methods and then conduct a single cell data analysis, this produces a much more successful result than if you were not to use AI and machine learning. This allows any artifacts to be removed. This allows for accurate clustering results, and it also allows for interactive and easy downstream analysis using those results that you provided. That's why AI and machine learning were kind of implemented in single-cell genomics in the first place. So now that we've kind of discussed how machine learning algorithms were implemented in single-cell data analysis and their ability to produce clear and accurate results, there are some challenges that still exist. One of the biggest challenges is that data sets are continuing to grow. Some data sets are reaching over 1 million cells, which clearly is pretty exponential from dataset sizes in the past. Not only that, machine learning frameworks that are implemented for single-cell genomics don't necessarily add speed. They produce efficient and clear results, but it's not going to increase any computational time, which clearly a lot of the attendees here are experiencing. They're experiencing a lack of machine power. And that also ties into another challenge, which is that sometimes researchers don't have the compatible machine resources that are required to process a very, very large single-cell data set. And with all these challenges occurring, as a computational researcher might seek solutions, increasing complexity can occur, which can result in a lot of mental barriers or apprehensiveness about attempting such solutions like we're going to see, GPU acceleration. They might be a little bit nervous about implementing it because of all the dependencies and requirements that are required in executing such a workflow. So to address all these challenges that we're seeing here, some independent solutions have been created and to address these specific challenges. One of them is implementing ultimately a faster workflow. And this is kind of where the NVIDIA part of the webinar comes in. This is involving using the same methods as CPU implementations, but just accelerating them using parallel processing in GPUs. So it's the exact same familiar single cell methods that are being used, just faster. Also, when using machine learning and AI methods in single cell clustering, it's required that it's a flexible environment to work in, that you're able to view the results clearly, review analytics clearly, and work in an environment that you're able to deploy and view models easily. And lastly is addressing the increasing complexity for the computational researcher, which ultimately you want to minimize. You want to ensure that a computational researcher is having a good experience in the platform that they're working on, and that they're able to really operationalize their team and work together. So these are the kind of solutions that we really need to focus on if we're to address these challenges that I mentioned. So today, well, what are we going to focus on specifically in these solutions? Today, we're going to focus really on the challenge of the increasing complexity for the computational researcher and how we can solve it to increase the enjoyment of the researcher experience and operationalize a team. So we're going to really focus on this in our solution. So how are we going to do that? How are we going to really create a researcher experience that'll be effective? Well, that involves several components that I mentioned, which I'm going to refer to as a stack. At the bottom is the foundation that I mentioned at the beginning, which is the implementation of AI and machine learning in sequencing analysis as a way to produce efficient results for growing single cell data sets. But the challenge still exists of computational time, which is the next level of the set, which is GPU acceleration. And we're going to address this by using examples from the open source NVIDIA Clara Parabricks toolkit, which is a suite of examples that is addressing GPU acceleration within single cell genomics using NVIDIA CUDA and the library of Rapids, which focuses specifically on GPU acceleration of end-to-end workflow. So we're going to implement GPU acceleration through NVIDIA. And the last part is adding that really compatible researcher platform that researchers can work on easily and operationalize their team. And that's going to be provided through the CodeOcean Compute Capsule. And this is a computational environment to package all the other components of the stack, the NVIDIA Clara Parabricks toolkit and AI and machine learning libraries to develop an all-around accurate environment for accelerated single cell analysis. So some of you may have heard of AI and machine learning libraries before. Some of you may have heard of NVIDIA before, but I don't think many of you have heard of the CodeOcean Compute Capsule. So it's important to address what exactly is in a compute capsule and why it's so beneficial for the researcher experience. And this boils down to three components that are pretty fundamental in any experience that a computational researcher might have for analysis. The first thing that a computational researcher might come across is the setup or the environment for a specific analysis. And I believe that CodeOcean provides a really, really nice UX for this, allowing the user to select any compute resources that they may require for their analysis and set up dependencies really easily, providing through base images or other libraries for which you can install packages. Once the environment is all set up, the computational researcher is going to focus on the code, actually editing and running their work. And once again, I believe that a compute capsule provides a really easy way to do this by giving the researcher multiple ways to execute models depending on their type of persona or their code proficiency, and providing a really easy way to organize, share, and reproduce their work, allowing, again, operationalizing of the team. And once the code is set up, once the environment is set up, the last part that's important to a computational researcher is being able to use their future analysis with other data that they might want to execute the analysis on. And CodeOcean Compute Capsules address this by providing, once again, a really flexible UI that makes it easy to work with large data sets, which allows you to upload from different sources. You can upload your data set from the cloud. You can upload your data set from your local machine. And this also ties into the computational challenge that we're facing in single cell genomics, which is growing data set sizes. It makes it really easy to work with those data set sizes in, once again, a really flexible environment. So these are kind of the fundamental components of a compute capsule that contribute to the researcher experience, and also what are going to tie into when we enter the demo. So now that I've kind of introduced the accelerated stack of AI and machine learning, NVIDIA, and CodeOcean, I've kind of explained what a CodeOcean compute capsule is, why it provides a flexible environment for the computational researcher. We're going to now get into two specific examples that provide an end-to-end workflow using this accelerated stack. We're first going to get into a compute capsule that contains the Rapids single-cell examples suite from NVIDIA Clara Parabricks. And this open source example is focusing on GPU acceleration on large single-cell datasets for analysis. The analyses that are conducted are on datasets from 60,000 cells to 1.3 million cells, which is clearly a very, very large dataset. And in this type of capsule, you're going to see some pretty typical analysis that are run, such as TSNE or k-means clustering. And these can be used to identify potential biomarker detection or cluster by cell type, which we'll see later. The second compute capsule that I'm going to get into is called ATAC-Works. And this is a deep learning toolkit in conjunction with Rapids and CUDA GPUs that address coverage track denoising and peak calling for low coverage ATAC-seq data. Now, what is ATAC-seq data? ATAC-seq data focuses on chromatin accessibility. It's data that can identify points in the chromatin that can be potential targets for either pathogens or other types of interactions. But what the problem is with low coverage ATAC-seq data is that it's very hard to determine what is just noise and what is actually a peak that is identifying chromatin accessibility. And in order to address this problem, usually you need tens of thousands of ATAC-seq cells. But with this type of deep learning toolkit, you can correctly identify actual peaks versus noise using just tens of cells, or even just a lower number of thousands of cells. Usually you would need maybe 20 or 30,000, but the example I'm going to show you is able to identify correct peaks with just 5,000. So these are the two capsules that we're going to look at today. Maybe I'll pass it to Sharon if anybody has any questions before we dive into the demo.
Yeah, so let's see, we got a few questions. I think Emily, you probably want to address some of these questions after the demo and after we have a better understanding of how it all works together.
Absolutely.
One question I did get that I think we can stop on for a sec is what machine learning frameworks can a compute capsule of CodeOcean support? Meaning, are we supporting only what you're going to present, or can we do a bit more than that?
Absolutely. So CodeOcean compute capsules provide a series of starter environments that the computational researcher can work on. And depending on the one that you select, it usually supports the majority of the popular deep learning libraries or machine learning libraries. For example, selecting a Python starter environment provides support for libraries such as PyTorch, TensorFlow, or Keras. So there really shouldn't be any issue when you're trying to access those machine libraries. So this is what we're going to get started on. This is a compute capsule, and I know it's probably very new to a lot of you to see this kind of platform in action. So we're going to walk through it one by one, and we're going to go into this through the eyes of a computational researcher, once again, kind of identifying what their experience would be navigating this platform. So first on the left here, you have what is a typical file tree system. You have folders that contain your code, your data, any environment setup that you may require, and also results. So now we're just going to go into metadata, which contains any description or information about the capsule that I'm showing you. So once again, this capsule is focusing on the Rapids single cell examples provided through the open source NVIDIA Clara Parabricks. It provides notebooks that are demonstrating how to use Rapids for GPU accelerated analysis of single cell sequencing data. You'll also see in the image here is a pretty typical the result that we're going to be seeing within the capsule once we dive into the actual code. It's a typical neighboring clustering, and in this case it's k-means clustering result of the data set, in which case each color is a cell type. So it's a pretty typical result that you're going to see in this example. And this provides any functionality status or any authors or research fields that you might want to add to further describe the capsule. And before I get into this, I should mention that both these capsules that I'm going to be showing to you today will be available on the public platform so that after this webinar, you can duplicate it and play around with it and see the impact that GPU acceleration can have on your analysis. And it's a really, really good teaching tool and an example. So before we dive into the code, like I mentioned, the #1 priority when entering an analysis for a computational researcher is the environment, ensuring that all the requirements and dependencies are met. And with these rapid GPU workflows, for example, they do require a pretty strict GPU compute capability in order to provide the correct amount of power and storage in order to completely run these workflows end to end. And this is provided through the environment tab in a compute capsule. As you can see here, you can change the compute service type, which allows you to select any machine that you require through AWS. It allows you to select any type of machine or instance that you may require. And you can also select it as a spot instance, which also decreases the cost when using this type of machine. But we're going to leave it as the regular for now. And it also provides a default machine, which is created upon the creation of the capsule, which is automatically selected in the creation of a capsule. So you have no problem there selecting the correct machine for this analysis. Also, another important aspect of the environment is ensuring all the dependencies are installed. In this case, Rapids does have a lot of dependencies and libraries and packages that have to be installed before any of the code can be run. And so the environment and CodeOcean compute capsules are set up using the deployment of a Docker-based image. And in this case, it's actually really convenient because NVIDIA Clara Parabricks provides a base image that already has all the dependencies for Rapids pre-installed. So just upon selection of this specific starter environment, I can already have all the dependencies required for the examples pre-installed upon startup, which really decreases any setup time that is required when I've actually entered the edit stage of Capsule. So now we've got the environment set up, we've got all the machines set up, we can start to jump into the actual code. There are multiple ways that you can actually run code in CodeOcean. A lot of computational biologists usually have their IDE of choice, which is provided usually through a cloud workstation in the compute capsule. You can select any of these IDEs or softwares to run and to edit your code. In this case, we're going to go with JupyterLab, which is best for running Jupyter notebooks and Python code, but you can also select from RStudio, the terminal, or MATLAB. So let's jump into JupyterLab now to look at these examples. So now we're here in JupyterLab. It's a pretty typical setup. It's nothing that you wouldn't have seen before if you've worked with JupyterLab before. And we're going to take a quick look at the notebook that we're going to be running. So this is the notebook that we're going to be looking at today, which is the GPU analysis, if it'll load, is the GPU analysis of 1.3 mouse brain cells. And this analysis really focuses on cell type clustering and biomarker detection. So as you can see, it's a pretty typical notebook. You're importing requirements, you're downloading data. And as you can see, you're going to get some results like this here, where you're going to perform TSNE on the very, very large data set, and you're going to end up with this. You're going to end up with a large clustering map that is clustering cells by cell type, and you have 34 cell types here, which is pretty large, and each color, of course, is a different cell type. Pretty typical result in single cell genomics. You also have here some expression clustering of genes in order to determine where a gene is most expressed. And as you can see here, what they're trying to come across is that the gene HES1 is more expressed in the genes where STMN2 is not expressed, and vice versa. So there's a little overlap, and that kind of leads to interpretation about differentiation or other potential cell type inferences. So as you can see here, we have more clustering, more clustering. It's really interesting, and I hope you enjoy looking at these results when you are able to duplicate and play around with these capsules. So if you want to edit and run it through here, you can run it once again cell by cell like a normal Jupyter notebook. But if you want to just run it, the notebook head to toe, and just collect the pictures for potential downstream analysis or potential presentation, or even if you're preparing this type of job to be run by a biologist who maybe doesn't have that much experience in code. I know a lot of you have experienced that kind of translating your analysis and results. The compute capsule also has a method for that, which is called the app panel. And this provides a way for users to run just a no contact, one click run that will run the notebook head to toe and produce the results right away for you. And this is really beneficial for those who maybe aren't in the computational biology field, who have a little bit less code proficiency, and allows for the translation of results much easier. So as you can see here, I ran the exact notebook that we were just looking at, and now in the figures folder, you're going to have every single figure that I was talking about, which is all the k-means clustering, all the biomarker detection clustering. You're going to have it all in one folder so that someone who isn't proficient in code doesn't have to dig through the notebook and figure out how to run the notebook in order to get the results that they want. And so once you're done all editing, running code, setting up the environment, the last thing you're going to want to do is be able to manage reproducibility in collaboration and potentially share with others. So it's really easy to share capsules here. You can share it with anybody that you may like with different privacy settings. And you can also look at any type of traceability or reproducibility here through the timeline, which provides any sort of activity that was performed on the capsule. You can see when someone ran something, when someone entered a cloud workstation, what results someone did, what files they changed, which really allows for complete transparency in activity, and also allows some insight into whether others were able to reproduce your same results. So before I move on to the next capsule, maybe we'll take a little bit of a break, pass it to Sharon, see if anybody has any questions about what I just showed, or maybe about the work itself.
Yeah, so one question we got is how does a capsule work with machine learning ops tools and again, in other platforms?
Yeah, for sure. So I'm assuming that when you're talking about machine learning ops, you're talking about maybe UIs or environments that allow you to monitor and view your results, such as maybe MLflow, if any of you are familiar with it. And that's actually really compatible with the Compute Capsule, depending on if you want to maybe implement it as a Jupyter Lab extension. You can implement it as an extension through the cloud workstation and access it really easily there. You can also set it up so that it's configured upon startup, and then you can view your jobs through other websites as well. It's really easy to connect those and ensure that your jobs and deployments are running smoothly. So now we're going to run into the ATAC works data. And I'm going to do a little bit less on the environment and manage, since I kind of explained that in Rapids, but we already have the environment here. It's the exact same setup. We have the required compute capability machines, and we already have the pre-configured environment with all the dependencies installed. So let's dive into, once again, the Jupyter Lab and examine what it's like to run and edit code for this example and what the example that I'm providing is. Load really quickly here. This one is focusing on single cell coverage calling and single cell denoising. So like I mentioned, it's a pretty typical Jupyter notebook. You're importing data, you're downloading it, you're importing any packages that you may require. There's some clustering that is performed just to get a kind of sense of the data that is present and just get an idea of what kind of data you're working with. And then you're going to kind of get the ultimate result is ideally something like this. You have the ATAC-seq data that was imported, which you can see above. The ATAC-seq data was then separated into clusters, and each clustering was run through denoising and peak calling. So you can see, for example, on the left here, we have the noisy coverage. There's a lot of really small peaks, which should be denoted as noise and not actual chromatin accessibility peaks. So you first have the raw coverage here. Now you have what is called the denoised coverage. And you can see all those really small peaks are now gone because those were denoted as noise, and they were removed because they don't have any significance to the analysis of peak calling. And now with all the denoised coverage that you have left over, with all the valid peaks you have left over, you are now able to correctly predict the peak callings and the actual points of chromatin accessibility. And this is a really interesting notebook and example, and I do recommend that you look through it if you're really interested in this type of stuff, because it is really interesting. The results are very fascinating. So this is the type of results that you'll be getting if you were to run this example. You can see it runs in 105 seconds, which for a data set of 5,000 cells is quite an improvement in computational runtime. And as you can see, it produces really nice visual results for anybody who wants to use it for downstream analysis but may not be familiar with deep learning toolkits or deep learning libraries, which I see was also an issue for some of the attendees. It was a challenge they faced. It's laid out really nicely in this Jupyter notebook and produces really nice visual images to view results in. So those are the two capsules that we are going over today to show the exact implementation of single cell GPU accelerated example, RAPIDS and ATAC-Works. And once again, these will be available to anybody through the public platform who attended, who is interested in viewing the exact implementation and interested in playing around with them at all.
So Emily, another question that we got from the audience is, let me see how exactly it was put out. So how does the NVIDIA GPU support get configured for CodeOcean? I'm not sure on what level exactly you're asking here. So we can definitely go further with a deeper level answer. But Emily, perhaps you want to take that.
For sure. So when it comes to actual setup and configuration, a lot of that is done through backend code and backend development. So it's really not a concern to the user. When the user enters a compute capsule and is attempting to configure the environment and configure the setup, pretty much all of that is available to you through a flexible UI, allowing you to just click the one that you need and click the dependencies that are required. It's a really easy UI to kind of set up and just provide a very flexible experience for those who are interested in the configuration. But any details and really technical specifics that are required in the configuration of these GPUs are managed through backend development and backend code. So that's really not something that a user needs to worry about, which is really, really beneficial when it comes to implementing a beneficial researcher experience. So now that we've kind of looked at these two compute capsules, I'm just going to do a quick summary of what we went over. So we went over two specific open source example packages that provide an insight into GPU acceleration of single cell workflows. We went over RAPIDS, which focuses mainly on cell type clustering and biomarker detection of large data sets. And we went over ATAC-Works, which is focusing on denoising and peak calling of low coverage ATAC-seq data using libraries from RAPIDS and also NVIDIA GPUs. And we also went over kind of the structure of the computational workstation through the CodeOcean compute capsule. Specifically in the compute capsule, we went over what might be beneficial to a researcher when experiencing this type of environment. The setup in a compute capsule is readily available for any AI and machine learning libraries, and it provides a really flexible UI to select your machine resources that might be required. You can also easily capture results through a cloud workstation, and it's really easy to share your gene expression or potential single cell analysis with others once the code is edited and run correctly. And lastly, the timeline provides a really easy way for researchers to monitor reproducibility and traceability when others are interacting with the capsule. So maybe one last time, I'll throw it to the audience. If anybody has any questions, feel free to type them in the chat or put them in the questions box.
Emily, I think we can move on and perhaps address the questions one by one. Is there anything else you wanted to cover, or can we wrap up here?
The last thing that I'm going to cover is maybe just resources. So if you're interested in the Claire Parabricks Toolkit, which is an open source package, you can refer to this link here on NVIDIA's developer page. There are also open source GitHub links to the RAPIDS and ATAC-Work examples that I showed if you're interested in looking at the scope of what is covered in those examples and also the requirements that are needed to run those end to end. And lastly, we provided a page for anybody interested in any CodeOcean resources. So these are 4 great links if you're interested in following up on any content that was discussed today in the webinar. Sharon, I'll pass it back to you maybe to wrap it up.
Yeah, so maybe a word on what happens next. We're happy to share the webinar recording with everybody who attended today. We can also share free access to the GPU acceleration example capsules that you have just seen. These are just a few examples. So if you'd like to discuss further additional use cases and GPU acceleration of your existing code, please contact Emily. Her email address is right here. Emily, if you can move forward to the next slide. Everybody can see your email address now. please don't hesitate to reach out to us or to request the demo on the website by the Solution Science team. Thank you so much, Emily, and thanks everybody who attended today. If you have any other questions, please feel free to reach out to us. On behalf of CodeOcean and our presenter, thank you and enjoy the rest of the day.
Thank you.